Julia Worcester, and I am going to be talking about the patterns of genetic diversity of the long-tailed ground squirrel in Mongolia. The species name is Urocitellus undulatus, and I also worked with Amanda Reyes and Professor Brian McLean on this project. This is my overall poster, but I'll be going further into it on the following slides. So this is a picture here of the long-tailed ground squirrel, Urocitellus undulatus. There is a data gap on the data sets for the species and other vertebrates within Central Asia. Central Asia has been highly impacted by past and present global change drivers. Those include glaciation, downward movement of permafrost, climate change, and overgrazing. Professor McLean and colleagues have been studying the species for a long time, and they found that there are two divergent lineages, Eversmani and undulatus, that are separated by geographic location. However, their phylogeny had low bootstrap support, and they wanted to look further into it. So what we did was study the highly variable mitochondrial control region. That is a non-coding area, so it does not code for a gene. That makes it ideal for molecular phylogenies due to it not being subject to strong selection. It is more likely to accumulate mutations. And like I said, it is highly variable. So the methods, here is a map of Mongolia and Russia. In the orange is the species range, and then in blue is where we got our samples from. So we did a DNA extraction from the museum tissue samples found in Mongolia and Russian mountain ranges, and they were from previous expeditions. Then we amplified the DNA for sequencing. So we did a PCR amplification of the highly variable control region of the genome using primers Control L and TDKD. We then sent the DNA for Sankar sequencing at NC State University. After we got the DNA back, we edited and aligned the sequences in the program Genius. Then we built a phylogeny with the control sequence by using RAXML on the Cypress Gateway portal. So before I go into my tree, I wanted to show the previous tree done by Professor McLean and colleagues. So they used the genes cytochrome B and cytochrome oxidase 1, and they found that there are two deeply divergent lineages of the Urocitellus undulatus species. So those are the undulatus subspecies, which is the eastern lineage, and then Eversmani, which is the western. So they found that specifically in the Eversmani, there was genetic diversity divided among mountain ranges. So as you can see by the stars here, those are mountain ranges. However, this was not completely supported as the bootstrapping support is low. And also the Uves mountain range, there was, as you can see by the asterisks, there was some up here in the middle and down below. So this is my tree. It's a maximum likelihood phylogeny. And so, as you can see, there is still the divergent lineages with the undulatus and the Eversmani. However, what we were looking for in the Eversmani, like the big grouping of the provenances and the mountain ranges, was not completely found here. So I color-coded it to make it look a little nicer and easier to read. For example, the Govi Altai, which is in orange, there is a big group of them here. However, there is some up top and spread out around here, as this can be seen for other mountain ranges as well and provenances. And again, there was low bootstrap support. So in conclusion, genetic diversity is slightly partitioned among different mountain ranges in Mongolia. However, there is low bootstrapping support, and the control region of Urocitellus undulatus may not be the best area in the genome to use for finding the species diversity in the mountain ranges of Mongolia. This is because the control region, or it can be because the control region has a shorter locus, so there is less to look at, and also it does not have the variability as hoped. There's also the possibility that there is population expansion of Urocitellus undulatus, so the spread of variability may not have a pattern, or at least not yet. So what's next? We can look at other regions in the genome to create a better genetic tree with correlation between species diversity and geographical region. Finally, I would like to acknowledge a couple people and companies I would like to thank the McLean Lab and the UNCG Biology Department for helping me do this project. I would also specifically like to acknowledge Professor Brian McLean, Amanda Reyes, and Amanda Weller. Finally, I would like to thank NSF who gave us our grant for this project. And thank you all for listening.